Good afternoon. It's now my pleasure to introduce our next fireside chat guest, Kinnari Patel, President and Chief Operating Officer of Rocket Pharma. So the format for the session is again a 25-minute fireside chat. And if you have any questions, feel free to type them into the question box and we'll try to ask them on your behalf. So Kinnari, thanks again for joining us today. And would you please start with discussing Rocket's strategic approach to manufacturing?
Definitely. Thank you so much for having me here. At Rocket, we are focused, as you know, on seeking gene therapy cures. And our focus has always been about the disease, the unmet medical need. And based on the disease of interest, we figure out the platform or gene therapy platform that's applicable. So we can have a direct mechanism of action or to the gene of interest. And so that makes us a little bit unique in the sense that we have both ex vivo lenti and AAV manufacturing approaches in-house at Rocket. We use our ex vivo lenti approaches for really diseases that are already in phase 1 and 2 of our programs are having phase 2 pivotal study readouts later this year. And our AAV program, it's kind of interesting because we were one of the first companies a few years ago to treat adult patients with AAV therapy, AAV9 specifically, we needed to scale it up. And to allow for scalability, we realized that quality and consistency in production of the product was so critical, and we couldn't take any chances. So we ended up bringing AAV manufacturing in-house. And this has helped us allow a lot of flexibility in how do we produce how many lots of products per year. And it allows us greater control and quality, which helps us think about our Danon patient population, our AAV platform is using to treat Danon program. And our goal is that manufacturing facility we built out in Cranberry, New Jersey, a little over 100,000 square feet, will treat 15 to 30,000 Danon patients across Europe and US upon launch. And so our approach to manufacturing is different. Ex vivo lenti CDMO, using commercial grade CDMO from beginning as much as possible, and with FDA and EMA buy-in. And AAV is really focused in-house in order to scale up for the quality and quantity we need to treat the largest market of our indications that we're pursuing, which is the Danon disease.
So can you expand a little bit on Rocket's manufacturing capabilities? For example, your thoughts about when it is appropriate to bring processes in-house versus working with external parties, and also how Rocket can reduce some of the risks or variabilities that are associated with partnered manufacturing that we've been hearing about.
At Rocket, we think really carefully about our manufacturing capabilities. I think this is because one of our founders, our CEO, had learned a lot when he worked on the Kymriah project at Novartis, bringing it from academic institution into Novartis. So what we've thought about even before submitting our first IND is how do we get a product to be commercializable? How do we have a commercial grade product from day one as much as possible, where possible? This way we can decrease the risk of comparability. And we like to do that in-house versus outside. You know, that's always a question we get, like, how did you pick ex vivo lenti to be outsourced through CDMOs and AAV in-house? Well, the philosophy was simple. In ex vivo lenti, the CDMOs that produce CAR Ts, and there are numerous CAR T products that are approved in the US and the EMA, we realized that it was de-risked. So working with CDMOs that are already commercial grade de-risked our ability to execute and have that quality product that we need at the cost that we needed. AAV scaling up to adult site production, we realized that process development and optimizations is definitely needed, and the quality analytics and quantity of the product needed is going to be such great level of scrutiny. And we as a field are figuring out the AAV manufacturing together. And because of that, we decided to bring that in-house. And our goal was to bring things in-house to decrease variability as early as possible. So for our Danon program, we brought this in-house during our phase 1 program. So our phase 2 study that can hopefully be pivotal in its intention will have that final commercial product from a facility that can support both EMA and FDA compliant production.
Got it. So we just talked about the differences in the approach regarding the ex vivo and the in vivo programs. Are there any cost efficiencies or scale advantages that arise from having both of those modalities in your pipeline?
Yes, I think so. And I'm a huge advocate for companies to bring both platforms in-house if they can. And some of them really comes down to having database of analytics, both for ex vivo lenti and AAV. So the testing that you do, whether it's for raw materials or drug product or patient capabilities, patient material, that analytics, when you think about potency assay or ddPCR, that same philosophy gets applied whether it's a CAR T or ex vivo lenti or AAV product. So we have efficiencies when it comes to analytics and testing. The second place is when it comes to plasmids. When you think about plasmids, whether it's 3 or 4 system, the production you need when you have AAV versus lenti, you still need certain plasmids to be produced. So we get cost efficiencies by having same CDMO produce them across all 4 of our programs. And the third is learnings that are more specific on the vector production for ex vivo lenti can readily be applied to AAV manufacturing, especially because we're doing suspension based. So those production and scalability that we can apply the learnings on goes a long way. The other thing that I think we value quite a bit, and I come from a regulatory background, is when you have multiple programs across ex vivo lenti and AAV, the interaction you have with EMA, FDA, and MHRA, you learn quite a bit of what's applicable across the platform. And when you have one meeting on one program, you can actually proactively apply those learnings across all of your programs. And I think that's the other benefit of efficiencies you can see when you think about clinical drug development and doing drug development efficiently. There are quite a bit of synergies between the two. And even the CMC FDA reviewer that we have on our ex vivo lenti program is also on our AV, right? So that makes quite a bit of a valuable thing to have a discussion, good relations built with the agency on.
Mm-hmm. Right, right. And so let's ask a couple of questions on the Xbibo approach there. Can you briefly walk us through that general process that's involved in making an Xbibo lenti therapy, and also highlight the challenges that are associated with professionalizing this type of therapy for clinical and eventually commercial use?
Yes, in layman's term, I think the way I can explain ex vivo lenti is we essentially take patients' own CD34 cells, and that is one of our key materials, starting materials for the drug product. We take this by doing apheresis at the clinical site or at the hospital where the patient's screened and enrolled. And then we send that product or collected CD34 cells to our CDMOs. For Rocket, it's done through CDMO. So we send it to them where they use that patient material and essentially insert the corrected non-mutated gene of interest using the lentiviral backbone to create a drug product. And this cell processing happens in ex vivo lenti. For us, it's a 3 to 4 day product production of cell processing that happens. So that gets done. And once the drug product is made and put frozen or cryoed, we'd send out the drug product materials or samples for testing at our third-party analytics that's centralized. And once we have all of the green lights on, safety parameters, as well as some of the potency and efficacy parameters, and we know it meets our parameters for release of the drug product, we take that drug product and ship it back to the hospital. And in some of our diseases, like Fanconi anemia, we don't use conditioning. So this product, within a week of collection of patient material, by Monday, Tuesday, we collect the cells, we manufacture Wednesday, Thursday, and Friday, we reinfuse it right back into the patient. For diseases like PKD and LAD1, those we use cryo products. What we do in that situation is we actually, when the patient is ready for the material and they come back to the hospital, we condition or create room in the bone marrow so we can put in the corrected cells back into the patient. And in either of the situations, the entire process takes a few days of hospitalizations before and after. And this becomes really not just, I was excited 15 years ago thinking about personalized medicine. This really becomes individualized medicines in ex vivo lenti. And it's beautiful to see it all come together and seeing the impact that it has on patients' own cells, which allows them to really diagnose the disease and then potentially have a curative treatment option for the diseases. The logistic and the challenges that happen when you think about ex vivo lenti of bringing all of these pieces in a beautiful orchestrated way is a challenge. But at the end of the day, when you give the patient their own cells back and they're doing well, it all seems worth it. I think to make it scalable in commercial setting, having centers of excellence for clinical is really critical. Having your CDMOs that you have a partnership with from the beginning and having that calendar and good project management that allows everyone to be coordinated together for the same mission of treating each patient as if they were the only patient being treated. And this is the therapy invented just for that patient, really makes the logistics and the challenges of ex vivo lenti something that can be overcome and de-risked.
Got it. That makes sense. And so I understand. So that type of scalability and the project management is some extent what differs this in the late stage clinical and the commercial setting versus some of the early efforts in the in the academic setting. Understood. And so then with respect to AAV, that's an entirely different system there. Some of the issues that we often hear about are empty to full ratios, not with regard to manufacturing and other elements of the process. So for your AAV process, what do you see as the key analytics to look for in regards to evaluating manufacturing that can determine the manufacturing success?
We think about the analytics that are most important to us is purity and yield. If we think about purity of the drug product being produced and the yield, it can help us determine how much product to give a patient safely. So that incorporates some of the full capsid ratios as well as how much you can utilize to treat a patient per kilogram. And I think that's really critical. The other thing we think about is how do you predict if a patient AAV product is going to be sufficiently effective in patients is really going to be things like potency assays. So what we try to do is develop functional potency assay as early as possible so we can actually do potency assay work on the drug product as well as eventually for patient activities. And we can predict what is going to be the therapeutic effect that this product is going to have in these patients. So we believe at this point that process optimizations we've done it within Rocket and other sponsors around the industry that are sharing their knowledge, we are going to be improving AAV production. But at this point, our North Star and our focus is really about purity and yield and consistency of production.
Got it. And obviously there have been a number of programs that have been advancing in the AAV space. Some have run into challenges. Can you discuss what you've learned regarding AAV manufacturing from others in the space?
We've learned that anytime you're in the forefront of innovation, there'll be challenges, right? What we've learned is there are less challenges when you use suspension production, especially when you're trying to scale up to the levels that we need to treat adult patients. We've also learned that having mammalian cell line just makes it a little less challenging. I think the precedents that are being gathered throughout the industry and sharing of the precedents helps us also figure out how to have more scalable, higher scale product, right? So right now, if you think about it, few years ago, only 200-liter productions were happening. Now it's 800 and 2,000 liters. If you can scale up your production and what's being learned around the industry of how do you do that efficiently also helps us figure out how to decrease eventually the cost of goods. So when we think about the Danon disease and treating 15 to 30,000 patients in Europe and US, we need to make sure that the cost makes sense of these gene therapy products being produced. And I think there are some learnings that we're applying as we have it. And I think there'll be other learnings in the field that we're looking forward to collaborating with not just the experts, but the health authorities and our other sponsors to figure out how to do this even more efficiently in the future.
And regarding analytics, I think you touched on this earlier, and I wanted to come back to it. To what extent is the analytics in regard to AAV gene therapy, is that an evolving standard or an evolving space? Are there areas that are new types of analytics that you could see becoming more important as we learn more about what's in an AAV drug product?
I would say the analytics are evolving, but I kind of think of them across the platform. What evolution we're making, we're incorporating across both of our platforms. So for example, 4 or 5 years ago, we were talking about qPCR testing, right? And now we've converted to ddPCR. There's those types of innovations that are happening that we can apply to have not only more consistency on the production of the drug product, but analytics to allow us to more apples to apples comparison. The other thing we've done is automated some of our cell calculations or cell dosing calculations by using machines like NC200. So it's more automated and less operator dependent. And I think those types of activities really help. And the other thing that we try to do, and more so because we have to do it, but also because we try to do it, is we try to have our analytics and potency assays and all of these things early as possible qualified, if not validated, during our phase 1 program. And this allows us to better understand the data of the drug product and to be able to better correlate that to the benefit risk that we're seeing in the patient.
Right, right. And so in terms of capabilities, you touched a little bit regarding the scalability and the capacity that you'll need to target the Danon population at launch. Can you discuss a little bit regarding your perspective on what that will entail for Rocket regarding scalability and the pace at which you'll aim to scale up your process?
At this point, we believe that the quality of the product we can produce for our AV program, we can do one shift. I talk in shifts, sorry. It's a very CMC manufacturing focus. We can do one shift across multiple clean rooms of upstream and downstream and have it, our facility be running 10, 11 months out of the year to supply sufficient drug product that's needed, right? And I think that's really critical. But if need be, one of the things that we're doing is our Cranberry facility that's a little north of 100,000 square feet, half of that is devoted to clean rooms. And these clean rooms are designed to be commercial grade to meet both FDA and EMA requirements. So our ability to shift a clean room from upstream to downstream is very readily doable. But the other beautiful thing is when you have an upstream manufacturing in AAV, you can actually freeze the product and combine multiple upstreams to do a single downstream. So there are multiple ways to produce a product in a more efficient fashion and to maximize the capabilities. The other thing we could do for AAV is have 2 shifts in manufacturing that happens. So I think at this point for the patient population we're looking at, we feel quite comfortable that our Cranberry facility can have the needs be met both for the FDA and EMA. Of course, in the future, when we expand to Japan and other regions, we'll have to figure out dual sourcing capabilities as well.
Right. And so one of the other things that we've been hearing about is regarding the potential constraints in experienced talent within cell and gene therapy manufacturing. So can you discuss how Rocket is positioned to recruit and retain top talent in manufacturing and how that ties into your capabilities?
Oh my God, yes. Would love to talk about that. You know, until recently, I used to interview every single team member that joined Rocket. And that was such a treat, right? And the reason we did that, and we still do that across the leadership team, is we interview every single talent because we're hiring not necessarily for the skills, but the potential and the cultural fit our team members have. We believe that a mixture of individuals with deep expertise in cell and gene therapy and or disease paired with individuals that have high potential, as long as the cultural fit is appropriate and they have the same mission, vision, and philosophy as Rocket has, we can succeed. We don't need a team of 2,000, right? I use this analogy when you do the biggest filings, and the smallest company I've ever been at was BMS. So when we did a new molecular entity BLA filings, it wasn't 200, 300, 400 people that worked on this filing and submission and approvals. It was really about 12 to 15 people. So our goal is really to hire talent based on a puzzle. So at Rocket, what you'll see is this puzzle analogy. What are the strengths and gaps that each of us have? And what do we hire for based on where our products are in drug development? Our mission is really simple, to provide cures to patients with devastating diseases. But the mission in its execution is really lofty because these patients have such horrible, devastating diseases that they don't have luxury of time. So we need to do this efficiently. And as much as I would love to say we can hire individuals with lots of cell and gene therapy experience, and they exist, as you know, this is a new platform. And everyone is jumping for the same talent. So our goal is to have a training program in-house at Rocket that allows everyone to learn together. And one of our values is curiosity and lifelong learning. So what we have is all of our new hires go through a 2-week training program. And whether they're a manufacturing associate or a regulatory or commercial person, they learn everything from analytics, manufacturing, regulatory to RFP process. How do you negotiate a contract? And so that deep dive training program that we give to every single Rocket team members happens every 6 months. We supplement that with breakfast and learns, attending conferences, and sharing knowledge across the base. Our philosophy is simple at Rocket. We believe knowledge is only powerful and only valuable if you share it with each other. So that's the culture we have. And if we do that right, the beauty is you recruit great talent, you help them develop and help them elevate not just themselves, but the industry as a whole. And if they love coming to work and feel rewarded and feel like they're doing something beyond the helping the greater good of humanity, then they'd want to stay here. So I think that has become our retention tool is our culture. We hire for the culture and we retain because of the culture. And the ability to make difference in patient lives is just a North Star that we focus on.
That's great. I think one other topic that you touched on a little bit, and I think has come up over and over again over the course of this summit, is the regulatory stance regarding cell and gene therapy. And again, this came up a little bit in our discussion earlier, but can you expand on how Rocket is navigating the regulatory side of cell and gene therapy manufacturing? And in particular, are you noticing any differences in standards as Rocket is planning to advance the AAV Danon programs to later stages?
FDA and EMA are learning quite a bit, just like the industry is. And as they learn and get more filings, INDs, BLAs, late stage, they have more data to make guidances available. We are in this ride together with them. So our Rocket philosophy is have as many conversations as possible with all the health authorities, EMA, FDA, and MHRA as frequently as possible and as transparently as possible. We have seen that FDA is more focused on some of the analytics and assay and CMC component, where EMA is more focused on the benefit risk of a drug product clinical data. So our philosophy at Rocket is let's focus on the most conservative feedback that we get in either of the regions. And if we go with the most conservative, we can actually develop a global study, a global program that meets the needs of both of the regions. So we can ideally get both products approved in both regions nearly simultaneously or in parallel as much as possible. And the beautiful thing about having 4 programs in the clinic, including 2 in the late stage phase 2 pivotal studies across Europe and US, is we have RMAT designation and PRIME designations for these products. So we have quite a bit of discussions with health authorities, reviewers, as well as senior management. And that really goes a long way in learning and applying our learnings, not just to our wave 1 product, but our wave 2 pipeline that's heading towards an IND. And we believe that we can come up with an efficient drug development platform. And to help the industry as a whole, the other thing that Rocket we're starting to do quite a bit is working with ARM organization, as well as bio and other advocacy groups that partner with health authority like the FDA to talk about more policy, white paper, and influencing the future of how do we do this as a collaborative, scientific, data-driven approach way. And so I think we take great pride in the fact that we work in an ecosystem of other sponsors, health authorities, and KOLs. And if we do this well with patients in mind, even if the standards change, your data could help make a justification of why a therapy could be approved as efficiently as possible.
Great. So in the last couple of minutes, I also want to get your perspective on what, if anything, might people be underappreciating about Rocket's CMC capabilities?
Rocket, we tend to be a team that just works on internal execution. So a lot of times we are not in the panels. I think today was the first time we participated in a CMC panel. We like to do everything in stealth mode and have clinical data speak for itself. So I think some of the process development, optimization, scalability, and amazing deep expertise we have in our passionate team at Rocket, the outside world is only starting to see the tip of the iceberg as we have more programs going to late stage commercialization, but also more programs entering the clinic. I think that will become more evidentiary. And we like to be a company that shares knowledge as much as possible to the outside world, and with our especially the FDA health authority, but also with our academic institutions, so we can work on future innovations. And our goal is to be humble and learn from each other and incorporate as early as possible all of the learnings we hear about. And I think those types of things, time will tell, right?
Yeah, absolutely. So I see we're just about at the 25-minute mark. So again, I'd like to thank you, Carrie, for your participation and for your discussion of Rocket's manufacturing capabilities for this first time.
Love it. Thank you so much. We're excited to be part of this. And I think there are quite a bit of synergies across the platforms. And I'm so glad there's more and more conversation about CMC, because CMC can really make or break how well you have a product be manufactured and how much it impacts the patient. So thank you for this.
Absolutely.